Good afternoon, everyone. My name is Maxine, as it was said, and I'm a postdoc in the Hayden Gephardt and Plovritas labs at Stanford. And my long-term research objective is to identify mechanisms that underlie cancer metastases, as well as mechanisms that underlie the observed disproportions of specific cancer types in Black and minority populations. One such type of cancer is triple-negative breast cancer, which I'll be focusing my talk on today. But my goal is to really apply these experimental techniques that I'll discuss to health disparities research in minority populations. Also, I'm very fortunate and really excited to have just begun my early K99 fellowship. So today I'll be talking about my planned research for my K99 phase and how it's preparing me for my transition to independence. So triple-negative breast cancer accounts for 10 to 20% of all invasive breast cancers. These tumors are defined by the lack of three receptors: estrogen, progesterone, and HER2. And why is this important? Because these are the three receptors that we use to target breast cancer. And without them, triple-negative breast cancer becomes a very deadly disease, which is the reason I'm interested in studying it. So triple-negative breast cancer is highly aggressive. It lacks targeted therapies, which leads to high rates of relapse and metastases. And it disproportionately afflicts women of African ancestry. So triple-negative breast cancer remains an unmet clinical need. I have a long-standing interest in triple-negative breast cancer. As a PhD student in the Sweeney Lab at UC Davis, my dissertation work focused on studying microRNAs and methylation in triple-negative breast cancer. My work showed that microRNA 127 could inhibit the cell growth of triple-negative breast cancer cells both in vitro as well as in vivo. I also showed that microRNA 127 could suppress the metastatic potential of triple-negative breast cancer cells in vitro, as well as curb their ability to infiltrate into the mouse lymph nodes in vivo. My work was the first to show a genome-wide analysis of microRNA 127 regulation, and actually the first study to show that intravenous delivery of a microRNA could actually reach tumors implanted in the mouse mammary gland, which was extremely exciting. I was also interested in looking at LIG-1, which is a potent tumor suppressor in breast cancer, and I found that it was significantly downregulated in triple-negative tumors. I went on to show that this downregulation was caused by methylation of LIG-1, and using CRISPR-Cas9 based technologies, I could guide TET1, which is a demethylation enzyme, to LIG-1 DNA, resulting in reversal of methylation and upregulation of LIG-1 expression. Also, by adding in VP64, which is a very strong activator of transcription, I could upregulate the expression of LIG-1 by 3 to 18-fold in triple-negative breast cancer cells and restore LIG-1's tumor suppressive properties. Both of these studies were really done at the level of the tumor cell and its intrinsic properties. However, we can now appreciate that tumor cells don't exist in isolation, but in fact, they're surrounded by a diverse array of other cell types, collectively known as the tumor microenvironment. And we know that cells in the microenvironment can impact disease progression. For example, fibroblasts have been shown to sustain tumor proliferation and suppress immune response. And tumor-infiltrating lymphocytes are associated with improved patient prognosis in triple-negative breast cancer specifically. And so my postdoctoral research is really focused on understanding how the tumor microenvironment impacts metastases. And one place that triple-negative breast cancer loves to metastasize to is the brain. So brain metastases are extremely common for late-stage triple-negative breast cancer patients, and it's associated with very low median survival, the lowest compared to other breast cancer subtypes. So there really is an urgent need to understand the biology of triple-negative breast cancer brain metastases in order to identify novel drug targets and improve treatment options for patients that are living with brain metastases. However, there are still some things we don't yet understand. We don't understand the tumor microenvironment of triple-negative breast cancer brain metastases, as well as how cells in that tumor microenvironment interact with normal brain cells like neurons, astrocytes, and microglia. We also don't understand whether the decision or the ability to metastasize specifically to the brain is based on communication in the breast tumor microenvironment long before metastases actually occurs. So to address these unknowns, we really must ask two separate questions. The breast-focused question of what features of the tumor microenvironment correlate with metastases to the brain, as well as the brain-focused question of what features of the brain metastases microenvironment correlate with disease progression and patient survival. And we now have some really new and exciting techniques to be able to study the microenvironment in human samples. And I've chosen two complementary techniques: single-cell RNA sequencing and multiplex protein-based imaging. So single-cell RNA profiling, because it allows us to identify what cell populations are present in the tumor microenvironment. And multiplex imaging, because it allows us to spatially resolve the cell populations that are present in the tumor microenvironment. And together, these two techniques will allow us to answer those two key scientific questions. So why spatial data? I think many of us can appreciate the importance of studying DNA, RNA, and protein expression in cancer cells, but I believe the power of spatial analysis is rapidly being realized. So with all the possible ways that cells can talk to each other in the microenvironment, we can hypothesize that spatial organization impacts tumor growth. And recent studies suggest that patient tumors with different spatial organizations have different survival outcomes. So my goal in this work is to really understand how the cell types that are present in the tumor microenvironment, as well as their spatial organization, impact metastases. But in order to do this work, we really need human patient samples. And so my lab has a clinical database of all patients treated at Stanford with brain metastases. And this can be narrowed down to patients that had a primary diagnosis of breast cancer. And as you can imagine, it's taken a lot of time and a lot of effort to filter through 713 patients. And I've spent the better part of a year acquiring all of these patient samples to build a tissue microarray, or a TMA, that we can use for RNA expression profiling and multiplexed imaging. And here's an example of a mini version of our larger array that we just recently constructed. And so we are extremely excited about this data set because upon completion, I believe it will be both the largest as well as the most diverse breast cancer brain metastases tissue microarray, with 254 samples from 149 patients, including 6 different tissue types ranging from precursors of breast cancer all the way to brain metastases and each of the steps in between. And to provide a little bit of context, the largest commercially available tissue microarray I could find contained 43 cases of brain metastases, of which only 5 of those were from breast cancer. And our TMA actually contains 20 times that number of breast to brain metastases. And you can see the number of other tissue types here. This TMA will also be extremely powerful because each patient sample has been broadly consented for research, which allows us to obtain detailed clinical data on each patient, which is rarely available when you purchase a commercially available TMA. So I'm really excited to say that we have collected all of the samples that we need for our patient tissue. And we've even just built a mini version of this array, which you saw an image of it on the previous slide. And this mini array contains matched patient samples. So meaning we have both the breast tumor as well as the subsequent brain metastases from that same patient. So I will be using our mini TMA for RNA spatial profiling. So this will help me identify what genes are changing specifically in tumor cells when they transition from the breast tumor microenvironment to the brain metastases microenvironment. I will then transition to doing large-scale multiplex imaging of our full tissue microarray using the 50+ validated markers we already have and any novel markers we might identify during RNA profiling. I then hope to validate my findings in external cohorts of patient samples. And I expect by the end of this work that we'll be able to visualize and characterize the tumor microenvironment, as well as identify markers that predict the potential of metastases to the brain. And as I transition to independence, I plan to really intersect this work with research in health disparities. So I've really spent the majority of my training learning to leverage the best experimental tools to study triple-negative breast cancer, from microRNAs to CRISPR-Cas9 technology, and now single-cell sequencing and multiplex imaging to understand the tumor microenvironment of triple-negative breast cancer. And most of these models are based on non-minority ethnic populations, despite the fact that there is a 2 times higher incidence of triple-negative breast cancer in women of African ancestry. Survival is 14% lower at 5 years for young Black women with breast cancer, even when there's equal access to healthcare. And Black women have a 2.6-fold higher incidence of brain metastases and a 10-fold higher risk of death from these metastases. So as an independent researcher, my goal is to really shine a light on cancer health disparities and study the biology that I think contributes to the disproportions that we see in African American women with triple-negative breast cancer. And I will do this by maximizing advancements via the equitable study of race and cancer, also known as MAVERICK. And the overarching goal of MAVERICK would be to bring the best translational science techniques and apply them to answering cancer research questions in minority populations. So my goal as an independent researcher is to really ask new questions about the role of the tumor microenvironment in the context of health disparities, and to be able to even revisit old unanswered questions using cutting-edge experimental techniques. So with that, I will end. I would like to thank my mentors, Dr. Hayden Gephardt and Plavritas, and special thanks to my wonderful undergraduate student, Brianna, who has done an incredible amount of work helping us identify our patient cohorts. Special thanks to all the teams that have helped put this work together, and to my funding sources. And thank you to the organizers and to the audience for your time and attention. And I'm happy to connect for questions or comments via email or Twitter. Thank you.